Understand how the proto-oncogene story happens, okay? How the cells start dividing. In one line, if I have to, you know, just sum up this whole story will be that cancer begins when a normal grow, divide, rest system loses its control, okay? A normal grow and divide and rest system loses its control. These proto-oncogenes are these accelerators. They get stuck on, okay? It's like a switch on, and these tumor suppressor genes that are the brakes, they get broken, and the apoptosis, the handbrake, right, the self-destruction thing, it stops working. So you have to have all these things, in this order. Your proto-oncogenes have to be upregulated, your tumor suppressor genes have to be downregulated or maybe inhibited, and the apoptotic system have to be just inhibited, okay, just to be stopped, right? So the story begins like that. We have something called as gene, right? And this expresses a lot of mRNA, right? And this mRNA forms something called as translated proteins, right? And this translated proteins, they go and they secrete proteins out, right? And these protein, they become growth factors, right? So these growth factor, they go and bind to the neighboring or the cell within, okay? It can happen within. So the growth factor goes and binds to something called as growth factor receptor. Every growth factor has its own receptor. For example, epidermal growth factor has something called as epidermal growth receptor. So this binding happens. Now, after this binding, gene makes something called as transmembrane protein, right? These proteins, amazing thing. Okay, so genes make these transmembrane protein, which makes these growth factor receptor, and then the binding of these two happen, the growth factor and the receptor. Now, after the binding, there is something called as tyrosine kinase. Now, this tyrosine kinase phosphorylate a receptor, right? And once this phosphorylation is done, the receptor undergo dimerization. Now, meanwhile, a third gene, a third gene of a proto-oncogene, here, this third gene, right? Meanwhile, the story is that this third gene produces a signal transducer protein. It's a protein, it's a signaling protein. Now, this signal goes back to the nucleus, okay? Right here, it goes back to the nucleus, okay? Now, what happens here is that once it goes back to the nucleus, now these signals the nucleus to start making some new transcription factors. Now, we have new transcription factor in the story. So this goes and makes new transcription factor, and this transcription factor is going to come and bind to these 2 and 1. I'll tell you what is going to happen. Okay, so now we have new transcription factors. Now these factors, they produce cyclin proteins, right? And we need these cyclin proteins to jump from one phase to another. Which phase I'm talking? I'm talking from M to S1 and to M2. These type of phases, in the mitosis, meiosis, these phases. So from one phase to another phase, we need cyclin protein. If we have upregulation of cyclin proteins, we can jump from one phase to another without any problems. But if we have downregulation of cyclin proteins, it means it's a problem for phase, changing the phase. So then these cyclins, they disintegrate after we change the phases, right? That's the concept. Okay, after what happens? Now these cyclins, they bind with something called a cyclin dependent kinase, right? Now once they bind it, this CDK is present in every cell, but it needs cyclins to work. Now normally what will happen that cyclin can, get disintegrate, but in a cancerous cell, they will not disintegrate. They will go and bind with cyclin dependent kinase. Now this cyclin dependent kinase, it get activated and it phosphorylate RB proteins. Now these RB proteins, right, they start something called as E2F for DNA synthesis order, right? They produce this DNA synthesis order, right? They make this order like order for new DNA, right? These RB protein are responsible for it. So if we had good cyclin quality, we would not be able to, just make unregulated DNAs, right? So CDK phosphorylate RB protein, and RB protein further starts E2F, right? Now, what the hell is RB protein and what is it? So this RB protein is called retinoblastoma protein. Okay, these are the brakes of the cells. Okay, in normal state, RB protein binds to E2F. Right, this E2F is a transcription factor as well. Now, when it binds to E2F, it is kept at an off position. Okay, off. Right, it's kept in an off. But when the cell growth signal arrives via cyclin CDK phosphorylation, this phosphorylate RB protein, and then this RB protein, this phosphorylated RB protein, it goes and it changes the shape and it let go of E2F. It's like, okay, you can go. Okay, now this free E2F, enters in the nucleus and turns on the gene for DNA synthesis, which is called S phase. That's S phase in detail. So these RB proteins, E2F, they are these breaks, and free E2F can make a lot of DNA synthesis. RB is the break. CDK cyclin phosphorylates. Phosphorylates means remove phosphate from ATP, and it releases that break. It releases that E2F binded with RB protein. Okay, so this is the whole story, right? So till now we understand the story, right? Now time for some cancer. Right, now imagine this was the whole normal thing where this cyclin disintegrate, and cyclin never binds to cyclin dependent kinase, and then the RB can never get phosphorylate, and then E2F is E2F just is with RB protein, and then E2F is kept in check. You will never have any sort of cancer. So it's time for some cancer. What happens here is that because of some of the proto-onco genes are mutated, either of bad luck or acquired mutation, there is damage to the DNA. Now, due to the mutation in growth factor receptor, now this growth factor receptor doesn't need any growth factor. Now, if there is if there is no growth factor needed, it means they can get automated. They're going to get automated. Once they are automated, they don't need any growth factor. So growth factor doesn't need any growth factor receptors. These receptors don't need any growth factor to give signal. So signaling is happening. So, or if growth factor doesn't bind to growth factor receptor, the receptor doesn't allow growth factor to go away. It can happen like this as well. For example, the growth factor comes and it binds, and it stays in this position. It never binds out self. It never goes out self. It's always stuck here, binded, or there's just signaling without even binding. It can also happen like that. So this is just proto-onco, getting messed up, right? So there is now in both cases there's a gain of function in the receptor, and maybe there was a mutation in the first gene in the group of these proto-onco genes, right? These are the group of proto-onco genes 1, 2, 3, 4, 5, 6, right? Okay, so there is upregulation of this receptor of growth factor. So there is this, this is one way. So there is this another way to make mutation. Ideally, for every gene, there is one copy, right? But pathologically, there could be some extra copies of the genes, right? It's called gene amplification. Now, this can lead to number of receptors, right? Upregulation of number of receptors. For example, normally you have 2 receptors. But imagine you have gene amplification. Now, instead of 2, you have 20. Again, the signaling will be a lot more than normal, right? So, gain of function of receptor here also seen. Sometimes there is so much amplification that this extra copy might be present in the cytoplasm as well, or inside the nucleoplasm. We call this thing extra chromosomal double minutes. Okay, so in short, there is point mutation that can lead to changes in the receptor, right? Or there can be gene amplification that makes extra copies of the receptor to gain function. Overall, the idea here is that there is some sort of gain of function. That's the idea here, right? There is another way also of getting cancer. Let's take B lymphocytes. Let's focus on chromosome number 14. Focus on these immunoglobulin genes, right? Chromosome number 14. We have some genes here called Ig, immunoglobulin gene. This gene in particular is responsible for making immunoglobulins, okay, which is present on chromosome number 14. Okay, so what happened here is that they focus on immunoglobulin genes, right? Now, on chromosome number 18, there is a gene called BCL2. Okay. Now, this is an anti-apoptotic gene. It is anti-apoptotic means it stays on forever because it wants to stop, like it wants to stop apoptosis, right? So this is a very aggressive gene. It's a very aggressive in terms of its nature. So we have two chromosome 18, chromosome 14. Now on 14 we have immunoglobulin gene, and on 18 we have BCL2. So when these genes are present in their normal position, they are normal on their normal chromosome, they are well regulated, right? But somehow after some radiation or after some mutation, the chromosome mixing happens. Now BCL2 find itself in a chromosome 14, right? Now the BCL activity goes up regulation, goes it's like nuts. The activity of BCL2 goes up really high. Now this B cell over survives, right? And it may make something called as follicular lymphoma, because the CH14, the chromosome 14, is a very active in its nature, and it has to make antibodies, right? So I told you this chromosome 18 is a very aggressive gene, right? It's a very aggressive chromosome. Now, if there is translocation of this gene called BCL2, it jumps from 18 to 14. Now, because chromosome number 14 is a chromosome that is responsible for antibodies, and we know we need antibodies forever, right? If we need antibodies forever, right? So this CH14 has to stay on forever, right? So somehow this jump here from 18 to 14. Now, because of this jumping, the BCL2 stays on. And we know BCL2 is an anti-apoptotic, right? So if this B cell survives, right, anti-apoptosis means it will stop the apoptosis, right? The cell that was supposed to die will not die now, right? Imagine. So you will have more population of these B cell, and it will lead to follicular lymphoma. I hope we understand this. Knowing this, back to our concept, right? We have something called proto-oncogenes. Shift these genes from normal to some chromosome, and we get something called as gain of function, right? So this is a B lymphocyte, right? And I hope we understand this. To summarize, the gain of function can be in three ways. Point mutation at some receptor, right? There can be gene amplification by increasing the number of receptors, and there can be gene translocation, for example, as what we see. The BCL2 jumps from chromosome number 18 to chromosome number 14, right? So it can be three ways to gain function. Okay, so now we have we know that we have two copies, right? One maternal, one paternal of chromosomes, okay? To initiate this malignancy, we need to get stimulation, right, in only one allele, okay? So even one allele, if even one allele is mutated, it will alter the phenotype of the cell, right? Now it depends what kind of allele is getting messed up, okay? Now such genes that can alter the phenotype of the cell despite the other allele is already normal, those alleles are dominant allele, right? So those are dominant. So proto-oncogenes are really dominant. You just have to get one mutation in one allele, because you have two alleles, right? One from paternal, one from maternal. Okay, so tumor suppressor genes, for example, the name is RB genes, which hold tight to E2F factor. So these genes are tumor factor genes, right? They tumor suppressor genes, right? They suppress the tumor. So these genes, RB gene, they make something called as RB protein, and we know that RB protein inhibit E2F from DNA synthesis, right? Now, amazing thing is that even if one allele of tumor suppressor gene it goes bad, the other allele makes enough RB protein to keep E2F in check. So it's like a compensation happening. So even if you have one allele at hit, one allele got messed up, you will have other one that will compensate into production, into overstimulation of that other allele, so that you have enough RB protein so that you have this E2F function in check, right? So both allele have to go bad for you to become malignant. So that's why we call tumor suppressing gene recessive, because you need two bad genes to undergo cancer, okay? Because one hit from the proto-oncogenes is enough to alter the phenotype of the cell. Now that is dominant because it's one hit is enough to get you messed up to change the phenotype of the cell. Phenotype is just how it looks. That's it. Okay. So some of the names of the proto-oncogenes are okay. So we have 6 kind of genes. The first is PDGF, platelet derived growth factor. Second is epidermal growth factor, EGF. Okay. And second is these can be PDGF receptors or EGF, epidermal growth receptor. Remember that genes that make growth factors are proto-oncogenes. Okay. Cool. Third is RAS genes, RAS. Fourth is ABL genes. They make something called as ABL protein. Now, these ABL protein, they also, act on tyrosine kinase, and this normally they are, tyrosine kinase are these tumor suppressor genes, and you have to have two hits on both of alleles so that, you get cancer. So this tumor suppressor genes, these ABL genes, they suppress ABL, and in patient with chronic myelogenous leukemia and acute lymphoblastic leukemia, in these patients, these leukemic cells have special chromosome called Philadelphia 22. Okay, now this ABL is normally present in chromosome number 22, right? Now this chromosome number 22 is named Philadelphia. Okay, that's it, Philadelphia 22, right? So this ABL is present on that gene. Now if some genetic shit happens and you have this translocation, the ABL jumps from 22 to 9. Now the point here is that the 9 thing, right? It jumps from 22 to 9. Now where it joins at 9 is that it's called something called as breakpoint cluster. It, this ABL gene, right, at chromosome number 22, it jumps and it translocates itself on this chromosome number 9, and it breaks this, and we call it breakpoint cluster, right? BCR. I'll tell you what. Now, unfortunately, BCR and ABL fuse together and a new hybrid DNA is formed. A new hybrid DNA, right? Now, just look at this photo. I hope you can understand this, right? Now, this is the time ABL is mutated, right? And now ABL makes tyrosine kinase, it stimulates it further, and the cancer cascade continues, right? So there is this drug that shuts off this mechanism. It's called imatinib mesylate. Okay, so that's the concept here. Okay, so that was fourth, the ABL, right? This is ABL gene is a proto-oncogene. It can get cancerous if it translocates itself from Philadelphia 22 to chromosome number 9, and it joins. It makes a hybrid DNA with another gene which is BCR gene. Okay, so fifth gene, what is that fifth gene? The fifth gene is MYC gene. Okay, MYC. This MYC gene, right? If this gene get amplified, this proto-oncogene proliferates, and it proto-oncogene become oncogene. This MYC gene become oncotic. Cell has high risk of cancer. Which cancer? Myelocytoma, the cancer of myelocytes of the bone marrow, right? Okay, so I hope we understand this whole proto-oncogenes story. Now let's talk about a little bit about RAS genes, right? The number 3. These are also proto-oncogenes. Story of RAS. Let's understand RAS cascade. Now RAS has a GDP until there is a GDP. Okay, RAS has GDP, right? Until there is a GDP, which is a duo. It's a low energy state, right? It's inactive. After phosphorylation, the RAS gets activated and the GDP converts into GTP, triphosphates, right? Now this RAS gets activated, right? And RAS later phosphorylates RAF kinase, and this further results in MAP kinases, and then MAP kinase stimulate back to the nucleus. Now again, RAS kinase goes to the GDP position, inactive, right? Now this whole thing happens, right? RAF activates MAP, and MAP activates this nucleus, and the new proteins are formed, right? Now after all this shit happens, is that this GDP position goes back, this GDP on activation gets converted into GTP, right? And after all this shit happens, right, whole this RAF makes MAP, and MAP makes nucleus, and nucleus makes further proteins. Hold this shit, after whole this thing, the GTP converts back to GDP, a lower energy state, right? So that's the normal story. Now, this inactivation is done by something called intrinsic GTPases. GTPases means it removes this phosphorylation. Now, it's an auto off button. Now, the twist in the story is that there is a tumor suppressor gene makes a protein, right? Now, this protein stimulates and it increases GTPases, this thing, right? So, it's an auto off, right? If it increases the GTPases activity, so RAS is now mutated, right? Now this GTPase is non-function because of the tumor suppressing gene protein, right? Now this GTPase is non-functional and RAS is activated forever, right? So what will happen? A lot of MAP kinase activity, a lot of MAP kinase will stimulate the nucleus, and a lot of proteins will be formed. Now the most common way to get cancer is RAS getting activated forever. This is the most common way. And if the GTPase's activity increase too much, the RAS will stay inactivated and there will be less growth. So you can get problems in two types. Either this intrinsic GTPase can be deactivated or can be activated. If it's activated a lot, it will just turn off the RAS forever. And if it's inactivated forever, the RAS will be activated forever. So that's the whole concept of RAS. Let's understand the phases, the GO phase and then cell. So let's try to understand the phases, right? So we understand the cyclin need, right? We need cyclin to jump from one phase to another. Which phase are we talking? We are talking about from G1 to S phase. We need cyclin. And if we don't have good cyclin, we won't be able to jump. If we have too much of cyclin, everything can jump from G1 to S1. Not S1, G1 to S. That's it. So we have G null, that is G0 phase. The cell is resting here. And then we have G1, that's the growth stage number 1. Here the cell has to proliferate, right? Growth factor binds with growth factor receptors. Now cyclin, is needed. Then cyclin goes to cyclin kinase, and then the whole story. And then RB protein let go of E2F, and then this is how it happens. So I hope we understand this whole thing. From G0 it goes to G1. From G1 it goes to synthesis, and then synthesis to G2, and then N, right? Okay, so normally phosphatases remove this phosphate from RB and E2F becomes inactive, right? Now here what happens is that human papilloma virus 16 subtype, it causes cervical cancer and it makes something called as E7 protein. Now these E7 proteins replace E2F with itself. Now E2F is free and now it's freed. So this is how it happens. This RB protein is supposed to bind with E2F. Now this HPV makes E7 protein, and then you will have free E2F. Now this will make a lot of genes, a lot of proteins. It will just synthesis a lot of synthesis. So that whole story was about proto-oncogenes. Okay, I hope you understand it. And if you don't, try to just maybe something is wrong with my explanation or something I have written which is wrong. So please tell me and have a nice day. Take care.